Hi there, students. In this video, we're talking about the cell structure, function, and organization. Now, now, the cell is defined as the smallest basic unit of life that is responsible for all of life's processes. So, it is. I want us to see the following words there that are very important, and they are the smallest. It is the smallest. It is the smallest and basic units of life. So when you say something is basic, it means it is fundamental, it is very important. So it says smallest basic unit of life. And it says that it is responsible for all life processes. So it means that everything that happens, that everything, every characteristics that is attributed with a living thing, that is attributed with life, is first attributed with a cell. Is that okay? It's first attributed with a cell. In fact, there is a theory that cell that levels of organization of life actually begins with the atom. The atom. So the atom. Atoms are the smallest particle of an element that can take part in a chemical reaction. So when atoms come together, they form molecules. They form molecules, and when molecules come together, they form macromolecules. So these ones, atoms molecularize, and then molecules come together to form macro, then macromolecules. So these macromolecules in biology are termed as organelles. So that's why organelles are basically made up of um, of molecules, of compounds. Is that okay? Different compounds coming together in different ways to make up a particular entity. So that is for macromolecule, which is in this case organelles. So when organelles now function together, they form the cell. So the cell is the functional unit of life. So if the cell is the one unit entity that is able to perform all life processes, so organelles work together, function together to form cells, and then cells organize themselves in different ways to form tissues. All right. So tissues organize themselves in different ways to form an organ, an organ, and then organs come together to form systems, and then the systems also come together to form the living organism, the organism itself, the organisms. So, and when different organisms come together, no, not different now, organisms of the same species come together in their number, work together, they form a population. Excuse me, they form a population. They form a population. All right. So, we said a population. These guys form a population. Then whenever we now have a population of different organisms, then many different populations will form a community. Community. So what a community is essentially a different organisms that different species of organisms that live together in a particular environment. So when these organisms now interact together with their environment, they form the ecosystem, an ecosystem. So an ecosystem is an interacting community of different populations. Is that okay? So that an ecosystem, different ecosystems form the biosphere, which is our Earth, or the layer of the Earth where living things exist. All right. So this is all we have for um, our. This is all we have for the levels of organization of life. Now we're going to continue into um, what the general description of the cell. Now it says we have a single cell is often a complete organism in itself, such as a bacterium or yeast. So in further videos, we're going to be discussing the different ways in which organisms can exist. A cell can exist first. A cell can exist as a complete organism. So, as an organism in itself, such as bacterium, yeast, euglena. When we'll be talking about characteristics of the classification of living organisms, I'm going to delve into this better. All right, so you're going to see that a cell can be a whole organism or can be part of a living organism, as we have in humans, or be so we are multicellular humans, for example, and it can also be a part, and it can also be a colony. Is that okay? Such as we have in Spirogyra. So we said colony in Spirogyra. I want you to take note of those things. Colony, that is right there. Well, we have colony in Spirogyra. Colony, we said in Spirogyra. All right. So, and then we have single unit, single organism, single entity, such as bacterium or yeast. And the smallest known cells are a group of tiny bacteria called mycoplasmas. Now, mycoplasmas, the word myco means the word myco refers to fungi. So, anytime you hear the word myco refers to the word fungi, that is plural of fungus. Is that okay? Plural of fungus, all right. So, or related with the fungi, the fungus family, the fungus kingdom, the fungi kingdom, all right. So, then we have some of these single-celled organisms are spheres as small as 0.2 micrometer in diameter. So 0.2 micrometer in diameter, that is their size. Some of them are as small as that. And as you know what that means, it is only about 0.2 times 10 to the power minus 6. So it means you cannot view it with even a light microscope. You view it only electron microscope. So a light microscope cannot view it. Only electron microscopes can be used to view them. That is how small some of these organisms can be. All right. So then, as an individual unit or bacteria, as an individual unit, that is bacteria, the cell is capable of metabolizing, it's capable of synthesizing, metabolizing, that is performing all life processes, including breaking down and building up processes. Metabolism is divided into two. We have ana and kata, anabolism and catabolism. So ana meaning building up, and kata meaning breaking down, breaking. So kata means down, ana means up. Right? Whenever you get the word ana and kata in science, that's what they literally mean. Right? Now, it says that it also can perform the function of synthesizing. Synthesizing means producing. So these organisms can produce on their own, making its own energy and replicating itself. So it can also replicate itself via what? Cell division. Cell division. And being a unit entity that way, it's going to be able to perform what kind of cell division? Mitotic. Now, of course, in its cell, the nucleus will perform meiosis. Okay, the DNA will be able to perform meiotic division. So that the way this the what's it called the in the nucleus of the cell, the way the chromosomes are replicated for sexual reproduction is by meiotic cell division. But if it's just to replicate itself by cell division, it's going to be a mitotic one, mitotic cell division. So that is the way the organism multiplies itself by mitotic cell division. But if it's an organism that undergoes sexual reproduction, it's going to be undergoing meiotic division. And remember, I'm going to link the video where I talked about meiotic and mitotic division down in the description below. Now, in a multicellular organism, cells become specialized to perform different functions um, through the process of differentiation, right? So you will know that the cell undergoes three stages in its life cycle. The first stage is cell division. So after division, a cell matures. So we call it cell growth. We call that stage cell growth when the cell matures. And then after cell has matured, then the cell differentiates. The cell differentiates. So having differentiated, then it can divide again. Having differentiated. So after division, the cell, um, no, no, no. In fact, this comes first. This comes first. So the cell first divides, then it differentiates. So it is after dividing that it can differentiate. In differentiation is the cell becoming specialized in its function. The cell becoming specialized in its function. Then after specialization, then it can grow and mature. Cell growth is the number three stage. So cell grows and becomes mature enough to what? To divide again. Is that okay? So that is the way the cell cycle goes. Then we have the assemblage of similar cells form tissues, and the cooperation between tissues in turn forms organs, which carry out the function necessary to sustain life of an organism. So that was what we talked about in the cell organization of life, right? So now, now the next one, the term cells was coined by Robert Hooke. It was coined by Robert Hooke. It was coined by Robert Hooke. So take note of that name. It's usually asked. So that's why it's called the father of cell, the father of cells. So and he did that in the year 1665, and he did that when he examined a thin slice of cork. Cork is basically the 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 back of a particular lesson. He said he used under a microscope and he observed the honeycomb structure. There's the back of a particular plant. So he took a thin slice of it, and what he saw was honeycomb like structure. Honeycomb like structure. All right. So that is the description he gave it. Is that okay? And he said it reminded him of the rooms in the monastery. So that room, that cell, that word cell actually has a Greek word, it's from the Greek word cellular, which means small rooms, small rooms, right? So, and so that was what premeditated him himself. Said it reminded him of small rooms in a monastery because he was a monk. No, it wasn't exactly a monk, but like monks live in the monastery, so he he has a knowledge of what a monastery, the rooms in a monastery looks like. So then the other scientist that is very notable in the discovery of cell is Anton von Leeuwenhoek. Anton von Leeuwenhoek. So there are many definitions, many spellings that you might see of that. This Anton von, it could be VON. So in my own year, I used Van, but I've seen in some definitions where some textbooks that use VON for this von. So Anton von Leeuwenhoek. So he made a further discovery by inventing his own microscope lenses that were more powerful than the microscope of his time. So he made a very uh, powerful microscope um, that was more powerful than the ones that were used um, in his time. And he was the first to observe the human cell with this and bacterial cell under his own microscope. So Anton von Leeuwenhoek. So that's it's a very important point. Remember that Anton von Leeuwenhoek was the first to discover um, human cell. He observed the human cell under the microscope and the bacterial cell. Now, the electron microscope was invented, which made it easier to observe organelles and other smaller biological structures. And now, the invention of electron microscope is um, is was pioneered by many scientists. Is that okay? group of scientists. But the basic thing here, the basic take home here is that it was the electron microscope that made viewing organelles and detailed structures even in the organelles. It made it easier and actually possible. All right. So the next one says the organization of living things. So we have macromolecules, which are our organelles, as I said. And we have organelles, which are macromolecules, which are synonymous to organelles. Is that okay? So these organelles, these macromolecules, come together in different ways, and we're going to see that when we observe the different organelles, such as mitochondria, vacuole, lysosome, ribosome, and the plasmid reticulum. So when we observe this, when we are going to be treating all of this, we're going to see the way these macromolecules come together and form various organelles. So now we have these organelles function together to form cells, and from cells we have tissues, and from tissues we have organs, from tissues we have organs, excuse me. So from tissues we say we have organs, and from organs we have organism. We actually have systems, sorry, and before the organism. All right. So we've gone over this before. The next one. Now let's talk about the scientists that made huge contributions to the discovery of cells. We have Robert Hooke, who discovered cells, who is called the father of what? Father of cells. And then Anton von Leeuwenhoek, who discovered the human cell, protozoan cells, and the bacterial cell. And Robert Brown discovered the cell nucleus. Albert von Kolika, who discovered the mitochondria and actually said it was the powers of the cell. So Albert von Kolika discovered the mitochondria. Jacob Schleiden and Theodor Schwann proposed the cell theory. So Theodor Schwann proposed cell theory. Jacob and they are actually attributed for two different things are attributed to them. You see, this Jacob Schleiden, Jacob his name is Jacob Matthias Schleiden. Is that okay? Or Jacob Schleiden. So the basic thing is that he discovered, um, he proposed the cell theory, and he was a German botanist. He was a German botanist, and his proposition was on the basis of the the plant the plant cells. Is that okay? The theories he proposed the plants, the cell theory that appertained to plants. So he discovered that plants actually had cells, and then proposed theories that were appertained. So there are five basic theories of the cells, and the ones that was appertained with plants rather were attributed to Jacob Matthias Schleiden and Theodor Schwann in 1839. So this one worked. Jacob Schleiden worked in 1838, and this guy worked in 1839. So, and he proposed that the animals, this guy, he was a German zoologist, this guy was a German botanist, and he viewed the animal cell. This guy worked since he was a zoologist, and zoologists refer to what study of animals. So, uh, he proposed that animals were composed of cells. Animals were composed of cells. All right. Another person is Camilo Golgi, who discovered the Golgi apparatus, and that's why it is attributed to. I mean, it was named after him, Golgi. Camilo Golgi discovered the Golgi body. Then Joseph B. Cavendish and Cavendish or something. I'm not very sure about that pronunciation, but just um, get the fact that Joseph B, this guy, Cavendish or whatever, and period J. Peletier. I mean, my apology to those of you who understand, who know how to pronounce this. I'm just trying to, I think they are French words. So they discovered the chlorophyll. They discovered the chlorophyll. I think they are partners, so working together. So they both discovered the chlorophyll. And another one that was not mentioned here is Felix Dujardin. Felix Dujardin. So Felix Dujardin was the one. He was a French biologist. He was first. Let me talk about the fact that he's a French biologist, and he worked in the year 1835. French biologist, right? Then, and he also uh, he discovered that the cells have a particular content, and he named that content the sarcode, which was later he named it sarcode, and it was later named protoplasm. So what we know as protoplasm by right now, proto means early life, early, and plasm means covering. Is that okay? So that um, um, plasm actually means I think living or so. So then, but proto means early. Is that okay? So um um the Felix Dujardin named it sarcode, and it was changed to protoplasm by the person known as Perkinje just 4 years after um, after he named it. So 4 years after this, I think that was 1839, right? So that is another scientist that I forgot to add to this slide. Okay, so now the next slide. Let's talk about the cell theory. All right. So then Rudolf von Virchow. We didn't mention it. We didn't quite mention it. So Rudolf von Virchow also made a nice, huge contribution. First, let's talk about the first three parts. It says that all organisms are made up of what? Cells. So then, and cells are the basic unit of life. We talked about that. So there is no life apart from the life of cells. So that is why we say that all organisms are what? Made up of cells. And they are the basic unit of life. And cells come from what? Previously existing cells or pre-existing cells that have multiplied. This was described by who? Rudolf von Virchow in 1858, who stated the omnicellular as cellular. I think it's acellular, not acellular. Omnicellular acellular. That is, all cells come from previously existing cells. All right. Then this is actually a question that could be asked that who postulated the idea of omnicellular acellular, that is Rudolf von Virchow in the year 1858. All right. Now, um, the modern cell theory. There are three additions which we call the modern cell theories, which are the fact that DNA is passed between cells during cell division. So that cell division occurs during this cell division, DNA is replicated and passed to the next generation. Now the next thing is organisms of similar species have the same types of cell, both structurally and both structurally and chemically. So because I am a human, every other human, those of you that are watching this video are going to have the same type of cells, both structurally and what? Chemically. It's going to compose of the same structure, the same chemical nature, have the same chemical nature and have the same structure. And then energy flows within the cells. So it's by virtue of the flowing of this energy within the cell that we are able to perform various energy requiring processes in life, in our daily living, right? And we know that it is the mitochondria that is the organelle responsible for the production or synthesis of this energy in form of ATP. We are going to get into that in detail. All right, so let's move to the next types of cells. Now we have the prokaryotic cells and the eukaryotic cells. We are single-celled or the prokaryotic cell. We have two types of cells, which are the prokaryotic cells and the eukaryotic. Prokaryotic and the eukaryotic. All right. The word pro means before. Is that okay? So, and carry on. The word carry on, which is the suffix there. Carrion. Carrion is the word covering. Actually, it's used to refer to the nucleus. Is that okay? So we refer to prokaryotic cells as before nucleus. That is, they have no definite nucleus. That's it. Then eukaryotic. The word eu means true, and carrion also means covering. So true nucleus. So it means they have nucleus, right? So it says that they are single-cell microorganisms, and examples are the archaea, bacteria, and cyanobacteria. So this is used to group this group of cells, or used to group classify organisms into three other domains. I mean, there are two other domains that are derived from it. We have the archaea and um, the bacteria. Is that okay? Archaea and the bacteria. So these are a separate domain. Then domain are now grouped into the kingdoms. Is that okay? Kingdoms. All right. That is in hierarchy of classification now. Now, we said they have no nucleus. I talked about that. They have no nucleus, but have region within the cell where the genetic material is freely suspended called the nucleoid. So they don't have nucleoid. In fact, anytime you hear of the word oid, something bearing oid, it means that, so this word oid means like. Sometimes it could be like false, as in like it's not the original type, but it's like it's like it. It's like a nucleoid. So you hear of word like nucleoid, um, nucleoid, which means that it is not nucleus, but it is a structure that looks like the nucleus. So nucleus is actually a membrane bound organelle, and that is what prokaryotic cells lack. They have they lack membrane bound organelles. So a nucleus is a membrane-bound organelle. The membrane that binds, that encloses it, is the nuclear membrane. So the nuclear membrane is absent. That's what we mean here. Essentially, that the genetic material is floating freely outside. It's just floating freely, the genetic material, the nucleoid. So you're going to have a structure like this, then you just see the genetic material freely suspended in the nucleus. Although it also has by virtue of the possession of, I mean, the reason rather that why this nuclear material is actually, or genetic material is coiled up this way, is because of the presence of histone proteins. You see, histones are proteins that help the DNA. You know, the DNA is coiled up in thousands of folds, even millions of folds. So histone proteins are the proteins that make this possible. Because without that, how do you imagine a a human cell possess DNA that is as long as that is meters long? So if something that is meters long is being enclosed in something that is as small as this, as less than a micrometer diameter, I mean, how do you think that is possible? It's made possible by the there are other things, other forces such as the dipole-dipole interactions, and other chemical chemical forces that help to bring this molecule together, these macromolecules together. But histone proteins also perform a major function in doing this work. Then the hereditary material can either be DNA or RNA, but never both. So, not like it's never both, no. It is for viruses. So, hey, I remember that. Viruses always have DNA, have only DNA or RNA. They never have both viruses, right? So, prokaryotic cells may have both. So that is why you see ribosomes float freely in their cells. We are going to have pictorial views of these things in a moment. But I just want us to, have understand some of these things. So ribosomes, which are basically produced by RNA, you know about the transcription, right? And translation. Transcription is where DNA turns to DNA, and translation is where DNA turns to RNA. Is that okay? Um, and when DNA is turned to RNA, um, when DNA is turned to RNA, that is the uh, RNA which will give rise to which is going to happen in the ribosome. Is that okay? So it's in the ribosome that proteins are now being synthesized. I hope we got that picture. Right? The thing is, I'm going to still make in-depth tutorials on these things that I've just explained here. Right now, we talked about the fact that the hereditary material can either be DNA or RNA. Then prokaryotes generally reproduce by binary fission. So what is binary fission? Binary, the word bi means two, so that is two fission. When you hear the word fission, fission means coming together. I know, no, there is fusion, I'm sorry. There is fusion and there is fission. Fission means separation, splits, fission. While fusion means marry or come together. Come together. Right. So they split apart into two. So a single organism is split apart into two, and we talked about that. I said we split apart into two, and you have two daughter cells. That is what mitotic division. Division. So mitosis actually makes this possible. Mitosis makes this possible. All right. So let's go further now. And another mode of reproduction is also conjugation. So this is pseudosexual. So conjugation is just it's not going to be exactly sexual. It's just like conjugation is also like a marriage, but is a special form of sexual reproduction. It's a special form of sexual reproduction. I'm going to delve deep also into these types of reproduction when we want to examine when we want to examine reproduction. All right. All right. So, um, the we said no nucleus, and okay, we've exhausted this slide. Let's go to the next slide. Um, so we have the eukaryotic cells. All right. Are eukaryotic cells are multicellular organisms. Are found in multicellular organisms, and examples include plants, fungi, protozoans, and animals. All right, so there is true nucleus. So I talked about the fact that that word "u" refers to true. U refers to true, and the size of the cell ranges between 10 to 100 micrometers in diameter. So you see that is very, very bigger than the prokaryotic cell. We said the prokaryotic cell is about 0.1 micrometers to 0.5 micrometers, while these ones are from 10 to 100 micrometers. Right. Then they reproduce asexually and sexually. So both sexual and asexual reproduction is possible. So you will find out that we can reproduce the cells, somatic cells can reproduce mitotically, can divide mitotically, and um, they can divide meiotically. Is that okay? So um, then sexual reproduction is now made possible because of meiotic division. Meiotic. I hope I spelled that well. Meiotic division. So they can divide meiotically, and those are non-somatic cells. Non-somatic cells means soma. The word soma means growing. Is that okay? Soma means growing. So cells that don't grow are the sex cells. So these non-somatic cells refer to what? sex cells. Alright. Then we have some the hereditary material can either be DNA or RNA. So the hereditary material can be DNA or RNA. So we usually have both the DNA in our nucleus, and even our mitochondria also has its own RNA. So we have the mitochondrial DNA. So we call it mDNA. That's mitochondrial DNA. Then RNA. So we have different types of RNA. We have about three. We have the messenger RNA, the transfer RNA, and the ribosomal RNA. Right. So these are the most important types of RNA, and they have their different functions. Right. We're going to be delving into it when we'll be dealing with cell um, replication and DNA replication and translation. So we have some contrasting features between plant and animal cells. For example, we have the cell, the plant, I think that's a grammatical error. The plant cell contains chloroplast, cell vacuoles, and plastids. So these are plastids are these are chloroplast is also a plastid, right? So central vacuole. So and this animal cell do not. So our cells are devoid of these things, central vacuole and plastids. We actually have vacuoles, but not it's not centrally placed. So I'm going to be showing you the diagram of the animal cell. So it's going to look something like this, but you see that it's the center that we have a vacuole that is surrounded by what is known as the tonoplast. The tonoplast. The tonoplast. Tonoplast is the vacuole membrane. Right. Then let's go to the next slide. Alright, good. So let's quickly observe this. Because of the longevity of the video, I'm going to be stopping here. And before I start discussing the organelles of the cell, in the next video, I'm going to be discussing the organelles of the cell. So let's quickly just talk about this and go through the diagrams. Now you see this. We have this guy here is the animal cell. This guy here is the plant cell. This guy here is the prokaryotic or bacteria cell. So both of this guy and this guy are both what? Eukaryotic. So that's why you will see that there is a membrane here. So eukaryotic means they have membrane-bound organelles. So and that there is a central nucleus. So notice this is the nucleus. This portion here is the nucleus. This whole portion here is the nucleus. And this thing here is the nucleolus. You can see it. Nucleolus is a special part of the uh, nucleus. So it's a specially stained part. So like when you stain the nucleus, it is stained differently, the nucleolus. Right? And that is where ribosomal RNA is being synthesized. And so which means that it's ribosomal RNA that gives rise to the protein structure. Ribosome is actually a protein structure. So ribosomes are said to be produced in the nucleolus. So we are going to see these wire-like structures inside. Those are the chromosomes. Those are the chromosomes. All right. So we said that these are membrane bound organelles. So this is going to be a mitochondria. So you see the mitochondrion, that is one. Singular is mitochondrion, plural is mitochondria. All right. So this guy also has it. Now what you will notice now is that the vacuole is almost centrally placed. Almost centrally placed. But one vacuole is just located at a portion, at an extreme end. Okay. So another difference between ours and theirs is the presence of okay, we've talked about those differences, which is the fact that they have plastids such as chloroplast, and we do not have those plastids. This chloroplast is the one that houses the chlorophyll. So when you know it is the chlorophyll that the plant uses to what? To synthesize its food, the chlorophyll. And do not call it chlorophyll 2, please. It is double L. This is double L. Why double L? Right. So the chloroplast contains the chlorophyll. And you're going to see that this guy has a cell wall while ours don't. So our own does not know cell wall. We just have our cell membrane. So they have a cell wall and then an inner cell membrane. So you're going to notice this yellowish boundary here is the cell membrane. Then this inner portion here is the cytoplasm that we also have. We have the cytoplasm. And we also have, so you see, we have pores that they don't have, cell pores. So we have pores, but they have plasmodesma, or one is called one is called plasmodesma, and many are called plasmodesmata. Plasmodesmata has many plasmodesmata, not just ma, has many. Why one is called plasmodesma, right? So those plasmodesmas aid in cell-to-cell communication. We're going to talk about their functions in the next video. But I want you to now see a very contrasting, uh, a very big difference between these guys and these guys. See, they have these structures called pili. Pili. One is called pilos. So many called pili. Where is that? It's okay. That's this pili. Is that okay? These projections. They assist in movement and also cell-to-cell communication, stimulus, response to stimulus. While it also has flagellum. Now, why it is also possible for these guys, animal cells to have flagella, but mostly, you see, bacterial cells do always have flagella. Then you are going to notice this very important difference, which is the reason why they are called prokaryotic cells. So I told you the other time that the nuclear membrane, there is no nuclear membrane, that the genetic material is just free floating. It's not bounded by a membrane. Is that okay? So you see this yellowish structure here is wound around the stored protein. That's why it's able to stay in there. Um, so that is that. And you're going to notice these red spots there, these red spots, these dots here, are ribosomes that are scattered around it. There's ribosomes scattered in the nucleus. So ribosomes are also scattered around, not nucleus rather, in the cell, in the cytoplasm. Ribosomes are also scattered around in our own cells too. Right. So that is one big difference. And another big difference between this animal cell and this prokaryotic cell is the fact that they have a cell wall which they have a similarity to this guy, but they have they also have plasma membrane, and which is our own cell membrane also, and they have a cytoplasm also, right? But they also have a mesosome. A mesosome. Mesosome simply means meso means middle, or like midway, just mid. Some means body, or soma as I discussed the other time. So it means a body that is growing inward, that is growing that is growing in the middle, that is growing in the middle. Right. So that will be it for this video. In the next video, we're going to be talking about the functions of the various organelles of the cell. Let me see that. So we're going to be talking about first the cell membrane or the plasma membrane. See you there in the next video. Thank you and God bless you. Thank you and God bless you.